So I'm pleased to have Dr. Maciej Czerwinski here with us today. Maciej received his Ph.D. from the University of Maryland at Baltimore Medical School in the Department of Pathology. He has been with XenoTech since 1999 and currently serves as a Director of Consulting. Maciej guides the development of products and services for the analysis of drug metabolism and protein expression. Maciej also conducts research on the drug-drug interactions between biologics and small molecule drugs and is the inventor of XenoTech's single-freeze enhanced pooling and thawing hepatocyte product, CryoStacks. It is my pleasure to present to you Dr. Maciej Czerwinski.
Good afternoon, everybody. Thank you for joining me for this webinar on drug metabolizing enzymes and transporters in non-alcoholic steatohepatitis, subtitled Research Application of Variants of Fatty Liver Disease, FLD Tissues. Aims and outlines of this presentation are as follows. First, we'll go through basic facts about fatty liver disease and more specifically non-alcoholic fatty liver disease, which will also lead us to a definition of non-alcoholic steatohepatitis, which is a stage of this progressive disease. We'll talk about drug metabolizing enzymes and transporters in this group of patients specifically, which will follow with the introduction for the collection of normal and diseased tissues, that is livers, in the Sekisui XenoTech Research Biobank. Fatty liver disease and non-alcoholic fatty liver disease. Significant history of alcohol consumption is basic elements of differentiation of these two diagnoses. Non-alcoholic fatty liver disease is receiving particular attention because it affects people who already modify their lifestyle to avoid known pitfalls of drinking. This disease includes the entire spectrum of fatty liver disease in patients who have no history of significant alcohol consumption. It encompasses steatosis or simple steatosis, progressing to steatohepatitis and steatohepatitis with cirrhosis. In this condition, liver contains more than 5% of fat, and this is fat in a macrovesicular form. In this condition, presence of hepatic steatosis with no evidence of hepatocellular injury in the form of balloon degeneration of hepatocytes and no fibrosis. We mentioned that fat is macrocellular because liver is also, of course, subject of microvesicular steatosis, which is not associated with NASH. Microvesicular steatosis is histologically different, especially in the sense of no distortion to the nuclei. It's present in a fatty liver of pregnancy, as well as a syndrome called HELP for hemolysis, evaluated liver enzymes, and low platelets counts. Also is present in Rey syndrome in the patients taking nucleoside analogs, tetracycline, or valproic acid. Microvesicular steatosis is also associated with congenital defects or inborn errors of metabolism related to fats and cholesterol. Non-alcoholic fatty liver disease, going again to the definition, there is no evidence of hepatic steatosis either by imaging or by histology. Histology from the chunk of tissue or from a needle biopsy is considered gold standard. There is application of many modern imaging technologies to diagnose this disease, although percent of finding varies greatly with diagnostic modality applied. In non-alcoholic fatty liver disease, There are no causes for secondary hepatic fat accumulation, such as, by definition, significant alcohol consumption or use of steatogenic medications or hereditary disorders. Significant alcohol consumption means, if you go to the earlier papers describing this condition, 20 grams per day per individual, regarding of the body mass of the individual. Person who estimates him or herself, or we learn it from next of kin, consumes less than 20 grams of alcohol a day, may be classified, if has the disease, as a non-alcoholic. Steatogenic medication that we have to exclude in this diagnosis include methotrexate, tamoxifen, corticosteroids, among others. Hereditary disorders, which are also need to be considered for exclusion, are such as hemochromatosis or alpha-1 antitrypsin deficiency. In the majority of patients with non-alcoholic fatty liver disease, there is an association with metabolic risk factors such as obesity, diabetes, specifically or more often type 2 with insulin resistance, and dyslipidemia. Non-alcoholic fatty liver disease is further histologically categorized into non-alcoholic fatty liver or non-alcoholic steatohepatitis, which is a progressive form of the disease. Non-alcoholic fatty liver is defined as the presence of hepatic steatosis with no evidence of hepatocellular injury in the form of ballooning hepatocytes. NASH, on the other hand, is defined as the presence of hepatic steatosis and inflammation in the acini or in lobular inflammation with hepatocytes injury, specifically in the form of a ballooning necrosis with or without different form of fibrosis. First description of NASH was provided by Dr. Ludwig in 1980 in a publication titled Non-Alcoholic Steatohepatitis, Mayo Clinic Experience with Hereto Unnamed Disease. So this is a relatively young pathology, if you will. Inflammation is thought to be a key component in the development of non-alcoholic fatty liver disease. However, there is no clear marker that would be identified as a predictor of the disease. The adipocytes-derived tumor necrosis factor alpha and apidonectin are being evaluated also for their role in non-alcoholic fatty liver disease. TNF-alpha is increased by oxidative stress and is a pro-inflammatory cytokine, whereas apidonectin may have an antifibrotic effect. And there are multiple studies that have demonstrated that an imbalance of these two cytokines is correlated with progression to non-alcoholic steatohepatitis. Ballooning necrosis, which is also part of this definition here, is a specific type of hepatocyte injury diagnostic of NASH. The essential elements of NASH diagnosis are illustrated on the following slide. This is steatohepatitis with fibrosis. This tissue is stained with Mason's trichrome, coloring collagen in light blue. Characteristics of NASH are macrovesicular steatosis, which you recognize macrovesicular steatosis as these large white circles where fixing of the slide removed fat. Further characteristic of this condition is lobular inflammation with polymorphonuclear neutrophils, ballooning necrosis, and fibrosis. Ballooning necrosis examples on this slide would be, for example, here, There is a good example here, and perhaps couple of them here. Ballooned hepatocytes are recognized by being 3 to 4 times increased size as compared to normal hepatocytes. They have centrally located nuclei undergoing karyolysis, wispy cytoplasm, and are present in the vicinity of inflammatory cells. As far as fibrosis is concerned in this field, there is fibrosis located around the periportal region that extends into the lobule in form of these narrow collagen bands. And this is an example of a pericellular or chicken wire fibrosis. In a progression of this disease, these bands will thicken and connect between different portal regions or portal to central region. There is a little bit of collagen staining around the central region, but this is not evidence of any pathology. When we discuss non-alcoholic fatty liver disease, please keep in mind that this is a spectrum of conditions that starts from a fatty liver, which crosses thresholds of 5% of accumulated fat in form of macrovesicular droplets, progresses to inflammatory disease, and later develops a serious and lethal condition of cirrhosis when many hepatocytes are lost, replaced by fibrous tissues, and their function are severely diminished. Prevalence of this condition. Most common chronic liver disease in US currently is non-alcoholic fatty liver disease. It's related to adolescent obesity, which had quadrupled in the past 10 years. It could be considered somewhat alarming that 1 in 10 pediatric patients in the US has this disease. This disease has been seen, according to cited sources on this slide, as early as age 2, with NASH cirrhosis seen at the age of only 8. Prevalence in the US for non-alcoholic fatty liver disease is estimated between 10 and 46%. It's lower worldwide, between 6 and 35, which is another indication that this is a disease associated with so-called Western lifestyle, characterized by high caloric intake, high level of fats, and shortage of exercise. NASH in the US is estimated between 3 and 5%. One slide of the pathogenesis of this condition is to illustrate that this is not exclusively a condition that develops in the liver by the liver itself. As I mentioned earlier, diabetes type 2, dyslipidemia, and obesity are metabolic cofactors. In 1998, they and others presented a two-hit hypothesis to describe the pathogenesis of non-alcoholic fatty liver disease, whereby insulin resistance contributes to steatosis and constitutes a first hit, which sensitizes the liver to oxidative stress, which is, according to this theory, a second hit, resulting in inflammation, fibrosis, and necrosis. Additional theories, such as multiple hit hypothesis, suggest other factors, including action of adipokines, more specifically reduced level of adipokines produced by fatty cells, and mitochondrial dysfunction that may also contribute to non-alcoholic fatty liver disease. At present, the precise factors leading to inflammation remain unclear and are various, with many metabolites, cytokines, inflammatory cells, and dysregulated processes such as oxidative stress and autophagy having been implicated. I would like to mention or recommend it to you good reference for pathogenesis of NASH by William Ereville, published in the International Journal of Molecular Sciences in 2014. As you see also from this slide, there is a large number of processes of molecules that can become drug targets. Drugs are being developed for multiple NASH targets. Also giving association of NASH with other conditions such as type 2 diabetes, dyslipidemia and obesity, these new drugs will be a part of polytherapy, therefore chances of DDI are high. Other imperative to study drug metabolizing enzymes and drug transporters in NASH stems from a need for safe and effective dosing of current medication. The strongest evidence for clinically significant dysregulation has been demonstrated so far for CYP2E1 and CYP3A4. There is a large number of animal models based on a diet and genetics, and there is also a fair number of studies characterizing mRNA, protein, or enzymatic activities obtained from frozen human tissues of different stages of fatty liver disease and different quality of tissues. In my remarks, I will focus on somewhat scarce clinical studies, which I consider be more reliable or most reliable, and I'll indicate in some places how their conclusions are supported by in vitro studies. First, I would like to go to CYP2E1. This is an example of a study in which clearance of a single dose of chlorhexazone in biopsy-diagnosed patients and matched controls has been analyzed. And chlorhexazone is a marker substrate for CYP2E1. On this graph, we have area under the curve of chlorhexazone concentration in plasma. Healthy controls are indicated by solid symbols, and non-diabetic NASH patients, the same amount of individuals, is indicated with open symbols. We can see that there is a significantly higher clearance of this drug in the NASH patients. In the same year, there was published another study that took advantage of liver biopsies taken from patients undergoing gastroplastic surgery and subsequent weight loss. In that group of patients, clearance of oral dose of chlorhexazone was also tested, and NASH was diagnosed by liver biopsies. In this graph, we see a relation between body mass index, BMI, and clearance of oral dose of chlorhexazone. Normal controls are indicated with solid symbols, and the BMI is between 20 and 25, which is desired range, while patients in this group are severely obese. The BMI ranges between, say, 45 and close to 100. It is well recognized that majority of patients which are seen for gastroplastic surgery have non-alcoholic fatty liver disease or NASH. So this is another example of clinical evidence for increase or induction of CYP2E1 in NASH. Increased fatty acids in the liver which come from the meal, dysregulated fatty cells or dying fatty cells, as well as from the de novo synthesis and insulin resistance, may contribute to higher level of CYP2E1 in NASH. In turn, lipid peroxidation and oxidative damage mediated by this enzyme may contribute to progression of non-alcoholic fatty liver disease to non-alcoholic steatohepatitis. I also need to mention mitochondrial presence of this enzyme in relation to one of theories that mitochondrial dysfunction is one of the drivers of progression of NASH. Overall, I would say that evidence for induction of CYP2E1 in NASH patients appear to be strong. We'll move to information available about 3A4. First, we'll go with the study in patients with steatosis. This is an association between non-alcoholic hepatic steatosis and hepatic microsomal enzyme 3A enzyme activity studied in frozen tissues. And steatosis was evaluated from the slides. On this slide, we see a relation between CYP3A, 6-beta hydroxytestosterone activity, and level of steatosis in the liver. Steatosis, for purpose of this graph, was categorized as none, meaning less than 5%, mild, between 5 and 33, and moderate, above 33%. Authors in this study also genotyped 3A5 because there may be consideration that genotype of 3A5 is a possible source of difference between the groups, but it was not found to be a contributing factor. Additionally, PXR mRNA is quantified. It varied up to 500-fold between different donors, but correlated poorly with the mRNA of CYP3A4 or CYP3A5 or CYP3A activity. And the quantification of CYP3A protein was done based on Western stain with antibody against CYP3A4, 7, but not 5. There is another study, or there is a study of CYP3A in patients with NASH. This really was a sideline observation because this is a study from 1998 by Weltman and coworkers that was focusing on CYP2E1, but CYP3A4 immunostaining was also conducted in liver sections from patients with NASH and compared with control liver sections. In regard to CYP2E1, authors' findings were in agreement with studies we discussed earlier, namely CYP2E1 induction in NASH. But in relation to CYP3A4, authors observed reduction in protein. There is another study, this time from the University of Eastern Ontario, that was conducted with biopsied human patients. Therefore, since diagnosis is done with a golden standard, there is no pitfalls associated with classifications of patients and collections of methods employed in preservation of tissue. These are of no concern in this study, which makes the study even more valuable. Phenotyping of patients in this study was done with a single oral dose of midazolam, which is a marker substrate for CYP3A, and also evaluation of 6-beta hydroxycholesterol, which is an endogenous biomarker for CYP3A activity. At the time of this study, CYP3A4 reduction in patients with fibrosis and non-alcoholic fatty liver disease with type 2 diabetes was known, but less information was known for fibrosis or NASH. This study also included genotyping for 3A4 allele 22 associated with reduced activity of the enzyme, PPAR alpha, cytochrome P450 oxidoreductase allele 28 associated with reduced activity of oxidoreductase, as well as other protein associated with increased fibrosis in the liver. mRNA analysis or immunoquantification of the proteins was not done for all of these genes. It was done for 3A4, but not for the other ones. General conclusion of this study is that NASH and fibrosis are associated with reduced expression and activity of CYP3A enzymes. The results of the study are summarized in the following graphs. On the left-hand side, we see plasma concentration of midazolam and how it relates to disease state, either SS, simple steatosis, or NASH. We see that there is a statistically higher concentration of midazolam in NASH patients which are unable to clear it due to the reduced activity of CYP3A. This is in agreement with finding that endogenous marker for reduced or for activity of CYP3A, specifically for beta-hydroxycholesterol, is also reduced by patients with NASH, which have lower ability to produce this metabolite. It's interesting that a patient with a simple steatosis also have a significantly reduced level of this endogenous 3A4 or 3A marker. Other observations of this study also relates to 4-beta-hydroxycholesterol, which is reduced in relation to fibrosis. Individuals with a non-alcoholic fatty liver disease, which may or may not have fibrosis, can be separated, and those with fibrosis have lower 3A4 activity. Also, in NASH, level of hepatic CYP3A4 mRNA is reduced, approaching level of significance. This study offers strong evidence of suppression of 3A4 in NASH and by fibrosis. Of course, it is or has been hypothesized that pro-inflammatory cytokines contribute to suppression of CYP3A4. In vitro studies, for example, with the pro-inflammatory interleukin 6, of many P450s evaluated, CYP3A4 is the one that is most sensitive. But if you recall what we said about the induction of 2E1, pro-inflammatory cytokines in vitro systems have also inhibitory effects of 2E1 suppression. So this is just to indicate that the picture is more complex than just action of pro-inflammatory cytokines. I also would like to share with you this reference slides for our further examination of the published data. Several CYPs were evaluated by Fisher and co-workers and study that is referenced multiple times on this slide. Let us focus first on CYP2E1 and CYP3A, since multiple observations are cited in this table, and we just discussed some other studies. In human studies, CYP2E1 activity was increased or induced in NASH. This appears to be supported by 4 of 5 studies listed in this review, with the exception of the Fisher. And Fisher was done with frozen chunks of liver obtained from multiple sources. CYP3A4 activity was found reduced in human studies, as we discussed earlier, and this observation seems to be supported by Weltman, but again, not by Fisher. This illustrates a difficulty to draw conclusion from mRNA, protein, and microsomal activity, draw from cryopreserved tissues that will correctly reflect in vitro human effects of this disease on drug disposition. Similar discrepancy can be observed in 2 studies referring to the effects of NASH on CYP2A6. Namely, mRNA increase was reported by Fisher, while decrease was found by Rubio. In evaluating findings of these and similar studies, it will be important to scrutinize inclusion criteria in terms of diagnostic tools of NASH applied to identify the patients, as well as what steps were taken to assure that quality of samples allows quantitative conclusions. Overall, effects of NASH on CYP enzymes are diverse, and formulation of a uniform guidance for dose adjustment may not be possible, at least at this time. Let us move briefly to membrane transporters changes in NASH, and I must start from saying that data on this topic is even more scarce. From this review table, if you will, a general conclusion can be drawn that uptake transporters are unchanged or perhaps downregulated in this condition, while efflux transporters are upregulated. There is a study on alcohol cirrhosis, alteration of nuclear receptor and drug transporters. In general, I would say that it appears that transporters are more difficult to study than well-characterized CYP enzymes. Regulation of transporters may have additional component of cellular localization, which makes it more complex to obtain protein expression data and view it in context of other changes, for example, mRNA level. I'll bring this study to your attention as somewhere among these samples, NASH samples may have been located, or alcoholic cirrhosis, at least histologically or pathologically in NASH or with alcohol, may be very similar. In this study, about 80 samples of human livers were evaluated by quantigen plex for mRNA levels, as well as for Western blotting. Samples in that study were categorized as normal, steatosis only, alcohol cirrhosis, diabetic cirrhosis, and diabetes. NASH as a specific category was not identified. And diabetic cirrhosis is alcoholic cirrhosis with presence of diabetes. In this study, conducted, as I mentioned, in a frozen liver, evaluated were the mRNAs and proteins by Western blots. In this figure, groups of patients are from left to right. Normal, steatosis, alcohol cirrhosis, diabetic cirrhosis, which is alcohol cirrhosis in patients with diabetes, and diabetes alone. Please note that NASH samples as such are not categorized here. Histologically, NASH that progresses to cirrhosis would look like alcohol cirrhosis if accompanied with diabetes would be most likely type 2 diabetes. Authors noted that several transporter mRNAs being different from control in alcohol cirrhosis, which is this middle group, but no alcohol cirrhosis with diabetes, which is a group next right to it. Uptake transporters being up or down regulated, and selected efflux transporters were upregulated. It's interesting that significant changes in the mRNA of these transporters are only noted in alcohol cirrhosis without diabetes. See, 1, 2, 3, 4, 5, 6 of them out of 9. Some nuclear receptors known to regulate CYP enzymes are implicated in regulation of drug transporters as well. In this study, in addition to transporters and CYPs, the mRNAs of some of the nuclear receptors were measured. In a simplified system, such as a plated primary hepatocyte, changes with the nuclear receptors and corresponding CYPs are expected to move in the same direction, but this is not the case in the samples from different stages of fatty liver disease. I highlighted some of these changes here. Based on the data in this study, the correlation between increased level of the receptor and the target mRNA is not present. It was observed that in a diabetic cirrhosis group, the pregnan X receptor PXR mRNA was decreased, but CYP3A4 mRNA was not different than control. So this is this group. While in a steatosis group, which is this group, CYP3A4 was increased without corresponding increase in the PXR mRNA. Similar observations can be made for expression of constitutive aldosterone receptor CAR and phenobarbital responsive CYP2B6 mRNA in this group of diabetic patients. In alcohol cirrhosis, the OATP1B1 mRNA was not different than in control, but the protein level of this transporter were increased in alcohol cirrhosis with or without diabetes. Similar apparent discordance between the mRNA and the protein was observed for some efflux transporters. This, in my opinion, may be caused in part by the methods used to collect and preserve the samples before the analysis. There is actually one more study in a pediatric NASH, although it's a preliminary pilot study. In children with NASH, which tend to have increased level of the acetaminophen glucuronide in systemic circulation, along with the increased excretion of this metabolite in the urine. A dysregulation of the hepatic membrane transporters MRP2 and MRP3 is a potential mechanism for these observations as postulated by the authors. Immunoblotting for MRP3 basolateral transporter in adult livers and immunohistochemistry for MRP2 canalicular transporter was provided as additional data. MRP3 was increased and MRP2 displayed altered localization. Liver biopsies were also conducted. As I mentioned, this was a preliminary study involving 9 patients with steatosis, 3 patients pediatric with confirmed NASH, and 12 controls. This graph illustrates findings of this study. On the top panel, we see that concentration of acetaminophen is not different between normal steatosis and NASH patients, while concentration of APAP glucuronide is higher in NASH patients. In this study, other APAP metabolite sulfate was also examined, but the findings were not remarkable. Going further, I'd like to bring your attention to one relevant study. This is the study of pharmacokinetics of atorvastatin in obese subjects. Regretfully, I must say that NASH is not named as a category of the patients, but look at the BMI and the comorbidities indicated in that study suggest that NASH patients are actually present there. In this study, OATP1B1, MDR1, and CYP3A4, which all determine dispositions of this statin, were evaluated. Uniquely in this study, paired liver and multiple small intestine sections biopsies were available. Therefore, this study does not have a normal healthy control group. BMI on average was very high, 45, ranging between 34 and 59. Insulin resistance or type 2 diabetes, hypertension, hypercholesterolemia, obstructive sleep apnea, and hypothyroidism were also present in at least 3 different patients, but unfortunately no data on alcohol was available. Authors of this study considered genotype of OATP1B1 and looked for allele associated with reduced activity of the transporter, namely 521 T2C change. Main conclusion of the study is that OATP1B1 genotype was a major determinant of atorvastatin clearance. Conclusions regarding effects of these conditions or NASH, specifically on these transporters, cannot be drawn from this study due to the fact that this was a fairly unique collection of biopsies not available from normal patients. Findings of this study are summarized, or some of these findings are summarized in these two graphs. We see that clearance of atorvastatin is related to amount of OATP1B1 protein found in the liver, and we see that clearance of this statin is also related to allele frequency responsible for active protein, that is allele T, and it's reversely related to presence of allele C associated with the reduced activity of the transporter. Further in this study, we see that hepatic CYP3A4 protein expression, which is on the left-hand panel, is reversely correlated with BMI, and the same is a novel finding for CYP3A4 present in small intestine. BMI was negatively correlated with the hepatic 3A4, as expected in non-alcoholic fatty liver disease or NASH, but also with the intestinal enzyme, and that may be related to hypothesis that circulating pro-inflammatory cytokines are responsible for suppression of 3A4. BMI was not associated with the levels of expression of OATP1B1 or MDR1 in the liver or in the small intestine. The lack of association of BMI with OATP1B1 and MDR1 proteins in the liver or intestine appears to be contradicting observations of the effects of NASH on these proteins reported by Clark and discussed few slides earlier, but seems to be in agreement with observations provided by Moore, also discussed earlier. Overall, findings in this study illustrate that discrepancies exist in conclusions on clinically meaningful effects of the non-alcoholic fatty liver disease and NASH on drug transporters. Again, overall, data on NASH effects on drug transporters is very scarce, and any recommendations about dosing of any drugs would be even more difficult than in case of cytochrome P450 enzymes. Let us move then quickly to the introduction of a research biobank. XenoTech processes many non-transplantable human livers into isolated hepatocytes, isolated Kupffer cells, and subcellular fractions such as Cis9, microsome, cytosol, or lysosomes. Support of basic research into liver disease is part of our mission. Tissues we accumulated over years may be of value for advancement of studies of human health. This motivated us to establish the research biobank and make these tissues available. Research biobank collects, characterizes, stores, and distributes human liver tissues. This is an example of one of our donors. Diagnostic features, donors with the non-alcoholic steatohepatitis. Diagnostic features are, of course, macrovesicular fat above 5%, lobular inflammation, fibrosis, and ballooning necrosis. This donor is 39 years old, male Caucasian, with BMI of 62.7 and macrovesicular fat of about 25%, without diabetes or history of alcohol use. The BMI and the macrovesicular fat content are not strongly related. Other parts of the body than the liver may be burdened with the load of fat. The microvesicular fat is also present, but fat droplets smaller than the hepatic nucleus is also present. When you look at the slides obtained from liver biopsies and compare them to slides obtained from major surgery, such as equivalent of a harvesting organ for transplant is, you may see one of many non-specific histopathological findings, such as indicated here in yellow, surgical hepatitis. In general, there are many non-specific histopathological findings seen in the liver biopsies. You may see some of them on our slides, and you'll see that the specimens are still classified as normal. These include mild and focal inflammatory infiltrates in the portal tracts that may be associated with mild portal fibrosis, that may be small focal areas of necrosis, and there may be often prominent Kupffer cells infiltrates. And of course things such as lipofuscin or hemosiderin inside the Kupffer cells are often seen. I'd like to go to definition of surgical hepatitis. It's interesting and somewhat new to me as well. So surgical hepatitis are clusters of polymorphonuclear leukocytes irregularly distributed in sinusoids, These clusters, so-called surgical hepatitis, most often are seen in zone 3 near the terminal hepatic venule in central vein or immediately beneath the liver capsule. It is thought that surgical hepatitis is caused by foci of anoxia within the liver or possibly by mechanical injury, perhaps from surgical retractors or just handling the tissue. Occasionally, true liver necrosis with rare acidophilic bodies formation can also be seen in surgical hepatitis, but there is no true hepatitis. Characterization of tissues provided for our samples and presented on data sheets include, of course, classification into normal, steatosis or steatohepatitis categories, and if they are other significant pathological observations, they will be listed on the top of a data sheet. Donor information include macrofat, age, gender, race, BMI, alcohol use, and if present, frequency or severity, and information about diabetes. Additional information is provided if hepatocytes isolated from given donor are present. Serological information is also included. Tissues typically are provided in a form of what we call pre-lysate. This is a chunk of frozen tissue stored in -80 that is dropped into the chilled buffer of choice and frozen and sent to you. So upon receiving, you potentially can retrieve the frozen fragment. Our data sheets also contain lower and higher magnifications of H&E stain of each specimen. For a reliable evaluation of macrocellular or even microcellular fat, low magnifications are preferred. Also, you can evaluate from those extent of inflammation, surgical hepatitis, or infiltration of Kupffer cells. A higher magnification is provided to illustrate features relevant to specific tissue. In this case, we have a normal tissue with a mild proliferation in the portal area. Presence of macrovesicular fat, of course, is noted. Inflammatory cells and Kupffer cells are present in sinusoidal spaces. You can also appreciate appearance of some glycogenated nuclei. Lastly, I would like to inform you or let you know that our bank contains adult and pediatric tissues. Currently, we have 53 adult specimens of normal tissue, 45 steatosis, some with and some without alcohol history. We have 14 cases of steatohepatitis, 7 of being NASH, and we have 26 cases of diabetes dispersed into these different categories. On NASH, I would like to tell you that our classification of a given specimen as NASH is very conservative. We don't necessarily go by this 20 gram a day, 20 gram of alcohol a day as a cutoff factor, because information provided by next of kin at the time of making decision about donation of organs sometimes indicate that donor had a sporadic consumption of alcohol or occasional, and we don't necessarily want to assume that this is most correct characterization of alcohol consumption of the donor, which is done at the time of a considerable stress of next of kin. So in our NASH classification, we only include those that are specifically described as no alcohol use. We have pediatric, 13 samples of them, and pediatric in our case in this classification is up to 4 years of age. We have other pediatrics going closer to age of up to 18, of course. And another thing I need to mention is that our inventory is expanding. Close to conclusion, I would like to say that it's our hope that the tissues that we gather can be utilized in further investigation of NASH, drug metabolizing enzyme transporters, biomarkers, or molecules investigated for their role in pathogenesis of fatty liver diseases. In case that information presented on our data sheets is not sufficient for you, please feel free to contact us. We'll find a way to provide you with access to existing H&E and Mason trichrom slides, and additional information may be available for some donors that describes their medical histories and hospital stay in more detail. I'd like to conclude my remarks and express my appreciation for your time and attention. Thank you.
Thank you, Maciej. As a reminder, if you have any questions regarding Maciej's presentation, please remember to send them via the questions pane on your GoToWebinar user console. Before we move into Q&A, I wanted to let you know that we are running a promotion on the pre-lysate tissue samples available through our research biobank as part of this webinar. Whether you order through our website or other means, you can receive 25% off US list price with promo code FEB25 in participating locations. This code also applies to our skin subcellular fractions in support of the new species that we are introducing this month. Current lists of promotions, as well as many of the donors available through our research biobank, are available on our website.
I see a question or two here. There is a question regarding, let me read it. Is there any research done / published on expression levels of uptake transporters? Well, I tried to scan literature for those that relate to uptake transporters, and I included them. So there may be some others that I missed, but there is not a abundance of publications in this area. If you go and carefully evaluate inclusion of patients, there are studies that are more generally in fatty liver disease or in liver disease in general, but there is a paucity of information on NASH. Can you make this bigger? This window down. There is a second question. Is there any information regarding the effect of NASH on enterohepatic recirculation? No, specifically on that, no. You may use information regarding, for example, intestinal 3A4 to draw some conclusions in cases of a specific drugs, but I have not seen any specific publications on that. Click on this one. Mm-hmm. Are there any studies done to investigate drug metabolizing enzyme transporters in laboratory animal models of NASH? If yes, is the trend consistent with clinical observations in humans? Yes, there is a good number of observations, and many of them, yes, are consistent with the human observations. I did not necessarily want to include animal studies. I wanted to focus on the clinical studies. and that's because when we realize complexity of a disease, it's understandable that not single model will fully capture human situation of this disease. Next question. Do you have liver microsomes and or hepatocytes from NASH? Liver samples along with 8 matched control samples. Hepatocytes from NASH, we may have maybe one in our history, so maybe not, but microsomes, yes, we certainly can make microsomes from the tissues that we have and offer them as a custom product. And that can be formulated into pools if such is desired. And liver samples with age-matched control samples, that will not be a problem. I think that's all questions that I had.
All right, if there are no other questions, this wraps up today's session. If you have questions regarding any of Sekisui XenoTech's products, please contact us by phone or email. For those located outside of North America, you can also contact your local XenoTech distributor. Thanks to all of you for taking time out of your day to join us, and thank you to Dr. Czerwinski for this wonderful presentation. Have a great rest of your day.